I want to start by thanking Diabetes UK for this wonderful opportunity to present the 2021 R.D. Lawrence Lecture. I feel truly privileged to talk about some of the work I've been involved with over the last 15 years. During that time, I've been blessed with working with many dedicated and passionate people working in the field of type 1 diabetes. I'm immensely proud to have been part of a true team science approach that has led to exciting discoveries that are changing and challenging the way we view this condition. Here is a brief overview of the presentation. Firstly, I'll tell you about a series of incredibly important and rare human type 1 diabetes pancreas biobanks. I will show you what the pancreas and the islets look like, and then through the use of some of these, the lessons we are learning about the immune attack against the beta cells, how immune cells recognize the beta cells, what might trigger the immune cells to attack the beta cells, and finally, if the islets are trying to fight back. At each step, I'll reflect briefly on how this knowledge could impact on type 1 diabetes treatment and management or prevention in the future. So as I'm sure you're all aware, type 1 diabetes is an organ-specific autoimmune disease where the body's own immune system targets the cells in the pancreas that produce insulin, called beta cells. These beta cells reside in the endocrine part of the pancreas in structures called the islets of Langerhans. The disease is believed to have arisen through a complex interplay of genetic, immune and environmental factors, which are still not fully understood. Much of what we know has come from studies examining blood taken from individuals with the condition. For example, we know that individuals with type 1 diabetes have evidence of circulating immune cells and antibodies that are directed against their own beta cells. And genetic studies have identified subtle changes in more than 50 genes that are associated with the susceptibility to disease development. Finally, a range of environmental factors have been suggested as triggers of an immune attack in genetically susceptible individuals. However, for us to truly start to understand this condition, we need to look inside the organ targeted in this disease, the pancreas. One of the key factors that has made this work possible is access to human pancreas tissue. However, this has not been easy. As you can see from the schematic here, the pancreas is buried deep in the abdominal cavity, which makes it very difficult to access. In addition, the pancreas not only produces the hormones that control blood sugar, it also produces many digestive enzymes. And for this reason, biopsies are not performed routinely due to high risk of complications. As such, the majority of pancreas available for research comes from individuals who sadly died with type 1 diabetes. However, this graph shows the cumulative number of pancreata available which have been collected and reported in the literature since the 1950s. Even now, in 2021, there has just been over 600 of these samples available for the research community to study. So what does a typical pancreas section look like? Following the collection of a pancreas, the tissue is preserved and cut into thin sections approximately 1 cell thick. This can then be stained to identify different cell types or molecules of interest using a process called immunohistochemistry. This is what a typical pancreas section looks like from an individual without diabetes. What you can see is a sea of grey-blue and these small islands dispersed throughout. The sea of grey-blue is the acinar or exocrine part of the pancreas. This makes up the majority of the pancreas and is the part that makes the digestive enzymes. The islands are the islets of Langerhans. I find looking at the pancreas like this reminds me of an island fort, and actually this is not so far from reality. So let's look a little closer at those normal healthy islets which are sitting in that sea of acinar tissue. When we do, we find they actually have their own defensive walls or a peri-islet basement membrane surrounding their outer edges, which helps to contain and protect them from that exterior environment. If we look inside the islet or fort, there are different communities of cells, each with their own specific set of roles. These different cells communicate with one another to ensure that blood glucose levels are controlled appropriately. There are specialized transport routes called the islet vasculature that allows fuel and oxygen to enter via the blood. And the products made within the islet, for example insulin and other hormones produced by the different cells and their waste products, can then leave the islets through smaller capillaries to be transported around the body as required. And finally, the islet also has its own form of power supply. Nerves enter the islet and can provide signals from the brain that can turn specific cells within the islet on or off depending on the stimulus received. I'll hope you'll agree that in a way the islets can be likened to a series of forts in a vast sea of hostile asana tissue. So what happens to these forts in individuals with type 1 diabetes? So here is a bird's eye view of a pancreas section from a recent onset EADB type 1 diabetes donor. We've used immunohistochemistry to stain for the insulin in beta cells, shown in pink, for glucagon in blue, a hormone produced by the community of alpha cells in the islet. And this allows us to identify the islets in which all the beta cells have been destroyed. And finally, in brown, we have stained for a molecule called CD45, which labels infiltrating immune cells, which are actively targeting and destroying the insulin-producing beta cells. By carefully looking at these sections, we can identify islets at different stages of beta cell destruction. So, for example, we can find islets where there appears to have been an early scouting party of immune cells near the islets, and this is called mild periinsulitis. We see islets where increased forces start amassing around the outer barrier, and this is termed periinsulitis. And invasive insulitis is where the forces have broken down that outer barrier and migrate into the islet to destroy the beta cells. Finally, we can see islets where there is a complete loss of one community, the beta cells. By looking at all the islets in a section, we get a window into the timeline of destructive events occurring in the pancreas of individuals with type 1 diabetes. By staining for different immune cell types, we can identify which cells are present in the most numbers at which stage of destruction. This information is critical for us to understand which cells we might want to target for treatment. In work that's been carried out over several years with these three talented postdocs, Abby Wilcox, Pia Leet and Rebecca Wyatt, we wanted to assess islet inflammation in type 1 diabetes. We did this by taking serial sections from our rare short-duration donors and staining with specific markers for the endocrine cells, such as insulin and glucagon, and for different immune cells using immunohistochemistry or immunofluorescence staining. We were staining for a range of different immune cell markers that include CD45, which you've already seen, and is a pan-immune cell marker. We also stained for CD4 to identify CD4 positive T cells, which can be likened to the generals of the immune system, telling other immune cells what they should do by releasing a range of chemical messengers called cytokines. CD68 to identify macrophages, which are like the cleaners who remove dead and dying cells, and CD20, which identifies B cells, and CD8, which identifies CD8 positive T cells. These two appear to have a particularly important role in type 1 diabetes. The CD8 positive T cells can be likened to the SAS, in that these are the immune cells that appear to be directly responsible for the killing of the beta cells. The role of the CD20 positive B cells is currently less clear, but they are thought to have a role in presenting antigen or small fragments from the beta cells to the CD8 T cells to promote their activation and killing abilities. Using recently acquired state-of-the-art imaging and analysis platform, part of UKRI funding for the Exeter Centre of Excellence for Diabetes Research, or EXCEED, and the unique EADB Pancreas Biobank, we have been able to identify a series of key age-dependent differences in type 1 diabetes pathology. These age-dependent differences relate to the type of immune cells present, the absolute number of immune cells present, the levels of beta cell destruction, and the processing of insulin. This started with an early observation by Pia Leet during her PhD, where she noted that there were a subset of donors who had evidence of many CD20 positive B cells in and around the residual insulin-containing islets, or ICIs. However, in a second subset of donors, these CD20 positive B cells were vanishingly rare. Two examples of this are shown here. where the CD20 positive B cells are identified by the brown stain and the CD8 positive T cells by the pink, with the islet shown in dark blue. For reasons that will become clear in a moment, Pia was able to divide donors into two groups, those with 3 or more B cells and those with fewer than 3 B cells on average per ICI. Nowhere was this difference more obvious than when we plotted the average number of CD20 positive B cells per insulin-containing islet against the age of onset of type 1 diabetes. If we look in donors diagnosed less than 7 years, 15 of the 16 had more than 3 B cells per ICI. In contrast, in donors diagnosed at 13 years or over, 29 of the 30 examined had fewer than 3 B cells per ICI, with those diagnosed between 7 and 12 being split between the two. It was what Pia discovered next, though, that really helped us to cement the idea that we may be dealing with two different forms of diabetes. Whilst examining the localization of the pro-hormone form of insulin, or proinsulin, shown in red here, and the mature form of insulin, shown in green, Pia found that in some donors, proinsulin and insulin were located in two separate distinct subcellular compartments. This is the staining distribution we typically see in beta cells from individuals without diabetes. However, in others, she observed far more yellow staining, indicative of co-localization of proinsulin and insulin in the same subcellular compartment. This suggests that in these individuals, insulin is being abnormally processed. Importantly, we were able to show that all those donors with a high number of B cells also had evidence of abnormal insulin processing, and those with a more typical distribution of proinsulin and insulin had fewer than 3 B cells on average in their ICI. This led us to propose that there are two diabetes endotypes, or forms of disease: type 1 diabetes endotype 1, or TIDE1, donors that have more than 3 B cells per ICI and evidence of abnormal insulin processing who are diagnosed young, and type 1 diabetes endotype 2, or TIDE2 donors that have fewer than 3 B cells per ICI and a more normal processing of insulin, and these individuals tended to be diagnosed slightly older. So although type 1 diabetes has one endpoint, in that individuals diagnosed with it will require insulin, perhaps it originates from two different disease mechanisms. Our findings suggest that there are two different endotypes, or forms of diabetes, and we have termed these forms TIDE1 and TIDE2. TIDE1 is characterized by a younger onset, many immune cells around the islet, which contains many B cells and CD8 positive T cells. There are fewer residual beta cells at diagnosis, and these are lost rapidly. And we also see evidence of abnormal insulin processing. In the TIDE2 donors, these tend to be diagnosed older, with many fewer numbers of immune cells around the islet, with very few B cells. And these individuals tend to retain their beta cells for much longer periods. So why does this matter? So these findings have implications for clinical trial design. Based on diabetes endotype, we could look to identify personalized and targeted immunotherapies. For example, teplizumab and abatacept, which target T cells, and rituximab that target B cells, are showing promise in trials. However, if we map that onto our endotype data, we see that the majority of individuals treated with these agents tend to be older and are likely to be TIDE2. We know that the TIDE1 donors have the most CD8 positive T cells and B cells, so we may hypothesize that these agents will be more effective in these individuals. And that is exactly what the efforts from the T1D UK Immunotherapy Consortium, Inodia and TrialNet are starting to address now. It could also have impacts for clinical management, as it suggests that individuals with TIDE1 need to be closely and carefully managed after diagnosis, as they are likely to rapidly lose residual beta cells. It highlights that for individuals with TIDE2, that we might want to look at strategies to preserve and increase the function of their remaining beta cells, because we know that preservation of even small numbers of beta cells is associated with better outcomes. Our vision for the future, though, is to find ways to determine endotype in individuals who are at risk of developing the disease, in the hope we can provide targeted treatments before clinical onset. So both TIDE1 and TIDE2 have evidence of CD8 positive T cells in and around their islets. And as I mentioned earlier, these cells are like the SAS in that they target and kill only the cells that they specifically recognise. This is one of the immunosurveillance mechanisms that the body uses to identify cells that are infected with virus or have become cancerous. To recognise these cells, they use a receptor on their surface called a T cell receptor, or TCR. Unfortunately, individuals with type 1 diabetes have CD8 positive T cells that mistakenly recognise healthy beta cells. In people without diabetes, these cells will have been deleted or inhibited. The T cell recognises the beta cells by interacting with another molecule called class 1 HLA. And this works by showing the T cell small fragments of proteins called peptides from inside the cell. If the T cell recognises the peptide presented by HLA class 1, this sends a signal activating its clinical killing mechanisms. Using confocal microscopy, we have shown that this HLA class 1 is strongly localised to the surface of the cell. This means that the CD8 positive T cells, capable of recognising beta cells that enter the islet, will be able to recognise and selectively target these cells for destruction. In individuals with type 1 diabetes who have CD8 positive T cells capable of recognising beta cells, this puts them at increased risk of being targeted. So I have described to you some hallmark features of a recent onset type 1 diabetes pancreas. This includes the hyperexpression of class 1 HLA in islets that still contain insulin, evidence of immune cell infiltration or insulitis, and the presence of islets that are devoid of insulin, where the beta cells have all been destroyed. So what is it that might set off the siege at the islet? And this is where environmental factors may come in. And in particular, a family of viruses called enteroviruses have been linked to the development of type 1 diabetes. But what is the evidence of their involvement? So as far back as the 1960s, enteroviruses have been linked with the development of type 1 diabetes. These viruses are very common and most people have a mild illness, but it can cause serious complication in infants and people with weakened immune system. However, there is mounting epidemiological evidence that points towards the development of islet autoimmunity following an enterovirus infection. A meta-analysis of 26 case control studies showed that individuals with type 1 diabetes-related autoimmunity were 3 times more likely to have evidence of an enterovirus infection, and this rises to nearly 10 times in individuals with clinical type 1 diabetes when compared to matched controls. The Coxsackie B members of the enterovirus family showed the greatest association. In keeping with that, persistent Coxsackie B infections from the TEDdy cohort was also associated with the development of islet autoimmunity in at-risk individuals, where children with consecutive CVB positive stools were 3 times more likely to develop islet autoimmunity than matched controls. Many of the genes that have been shown to be associated with type 1 diabetes risk are known to be expressed in beta cells, and these have been shown to be involved in pathways that sense and respond to viral infection. Importantly, these culminate in that class 1 HLA expression I've been telling you about. And we've also shown that enteroviruses, particularly from the Coxsackie B family, can infect human islets. So here is a section from an individual neonate individual who sadly died of a lethal Coxsackie infection. And when we stain for enteroviral VP1 or a viral protein, we can see that the islets light up. In addition, in vitro studies, if we isolate islets and infect them in vitro, we can see that there is clear evidence of infection. So we've shown that enterovirus infections are associated with type 1 diabetes development and that they infect beta cells. But can we find evidence of enterovirus in the pancreas of individuals with type 1 diabetes? To assess this, we use assays that can detect evidence of enteroviral genome or protein. And there have been several studies that have detected evidence of genome and protein in a range of pancreas biobanks, with a tendency of higher detection in donors with diabetes. The signal is, however, weak, with what appears to be just a few cells infected, indicative of a low-level infection, very different to that which we observed in those Coxsackie-infected neonates. So in summary, we have found no evidence of an acute viral infection in the pancreas of type 1 donors. However, we have confirmed the presence of enteroviral protein by immunohistochemistry, electron microscopy, and unbiased proteomics. We can show the presence of enteroviral genome confirmed by PCR, single molecule FISH, and EM, but not RNA-seq. Evidence of host response to virus was also present, and we had good correlation between the presence of viral genome, protein, and HLA class I hyperexpression. There was a high concordance across laboratories and assays in individual donors. And unlike in the non-diabetic donors, pancreata from type 1 donors with residual beta cells had multiple markers of viral infection. And these findings support the existence of a low-grade, recurrent or persistent viral infection that affects beta cells. So what might the implications for type 1 diabetes be here? Well, one option is to try to prevent diabetes development by immunizing individuals at risk of developing diabetes with a vaccine against enterovirus to try to stop that initiation of the attack on the beta cells. The data collected to date has been sufficient to convince a company called Vactech, in collaboration with Prevention Bio, to produce a CVB multivalent vaccine, and a first-in-man safety study test started in December last year. We could also look at treatments that could slow progression, asking whether antivirals given at onset of diabetes could slow the destruction of the beta cells. And a Norwegian trial to test this began recruitment in May 2019 and should be reporting back soon. So finally then, I want to ask if the islets are fighting back. So I've shown that in diabetes, the beta cells have increased classical HLA class I expression, which makes the cells more visible to the beta cell specific autoreactive CD8 positive T cell and resulting in their demise. However, those same signals that induce HLA class I expression can also increase the expression of non-classical HLA molecules called HLA E, F, and G. These non-classical HLA class Is can actually provide inhibitory signals to immune cells to turn them off. So there appears to be negotiations ongoing during the siege between the beta cells and the immune cells. There are those that are detrimental to the beta cells and those that are protective. Researchers in the UK and around the world, including Martin Eichmann, who recently joined the EXCEED team in Exeter, are working to identify ways to reduce the detrimental signals by, for example, selectively targeting the beta cell specific autoreactive CD8 positive T cells. If they can achieve this, we could tip the balance in favour of the protective response to preserve beta cells. So I hope through this lecture I've demonstrated the vital importance of pancreas biobanks to the understanding of human type 1 diabetes. These precious gifts are helping us to change the way we understand, will treat and potentially prevent type 1 diabetes in the future. I've shown how we have learned that type 1 diabetes is not the same in everyone, and this knowledge will help towards more patient-tailored therapies in the future and inform clinical management pathways. I've also shown there is clear evidence that enteroviruses can infect pancreatic beta cells, that they are associated with the development of type 1 diabetes, and that we can find mounting evidence of a persistent low-level infection in the pancreas of individuals at risk of and with type 1 diabetes. This has helped to pave the way for a vaccine against a subset of enteroviruses, and this is now in phase 1 safety trials. Finally, I've presented evidence that all is not lost in the islets of donors with type 1 diabetes. There is evidence that they are fighting back against the immune cells, and if we can develop strategies to tip the balance in favour of a protective response, for example through specific targeting of those beta cell specific immune cells, we could preserve beta cells. And we know that maintaining even small numbers of beta cells is beneficial for helping individuals to reduce the risk of complications, to increase time in range, and to reduce the risk of hyper and hypoglycemic events.